So assalamu alaikum. Today we're gonna start the microscopic features of the gingiva. Okay, so let's start it. Bismillahirrahmanirrahim. So first, the microscopic features in gingiva. You should know that from the composition of the gingiva. So gingiva is composed of two things. It is stratified squamous epithelium and the central core of connective tissue. And connective tissue, then it is they have said that epithelium is cellular. Matlab it contains a lot of cell in nature as compared to connective tissue, which is less cellular and is composed of the collagen fibers and ground substance, which is basically matrix. Okay, moving towards now, number one is the gingival epithelium. The gingival epithelium is the epithelium that surrounds the gingiva. So we generally aspects the general epithelium biology. Okay, so here we have that the epithelium compartment is thought to provide all so in historically the old people used to think that the epithelium just has to provide the physical barrier to infection, like it is a physical protection against any infection or any underlying gingival attachment that can attach to gingiva. Okay, however, now it is believed that it can play that has a lot of roles. That the gingival epithelium is not just a physical barrier; it is also an immune host defense by responding to a bacteria. Means when the bacteria enters or when it attaches to a gingiva, its innate host defense mechanism is activated. Okay, which means the epithelium participate actively in responding to an infection also. The next is signaling further host reactions, mean it will it will activate other host reactions or cells in the body to protect the body or to protect the gingiva from any harmful pathogens. And in next we have integrating innate and acquired immune responses. Well, it will integrate the innate and acquired immune responses. For example, now they have given the example how it will come across it. Epithelium cells may respond to bacteria by increased proliferation. The epithelium cells will be how it will start its division, proliferation. Next, in the cell signaling events, the cell signaling events will be there will be alternation in the cell signaling events. Cell signaling events is like the events like you first you will activate one cell, then you will activate another cell, so those events will be changed. Then there will be changes in differentiation. Differentiation is how much it is the extent of resemblance to the normal cell. And then the cell death will occur, and ultimately the alternation of the tissue homeostasis, as you all know about this. Okay, so basically it is important to understand the basic structure and function, as you all know. So they have given this box, so you should read it. They have give the functions of gingival epithelium. It has mechanical, chemical, also water and microbial barrier signaling functions. Then it has what things are involved in the architecture integrity. Major cells are keratinocytes. Other cells they have also discussed. Then the constant renewal, matlab the constant rate, they have discussed. Cell to cell attachment, how the one cell is attached to another. There are materials or there are things that are involved. Then how the cell basal lamina is. So here now starting the gingival epithelium, what it basically it has we have studied, it is stratified squamous epithelium. Now coming towards that, it is basically there are three different areas that can be defined by like morphological and the functional point of view. You have or outer epithelium or oral epithelium, cellular epithelium, and the junctional epithelium. These epitheliums are named on their morphology and the function they perform basically. Okay, moving towards the most important, they have said the principal cell type of gingival epithelium is keratinocyte. If you have been asked in your viva or in your papers or MCQs that which principal cell is in the gingival epithelium, you should know that is keratinocyte. And non-keratinocytes are Langerhans cells, marker cells, and melanocytes. We will read these one by one, these cells functions in the next upcoming videos. The main function of the gingival epithelium is to protect the deep structures. Yes, the gingival epithelium has to protect the deep structures. In deep structures, you have the main, because it can you have to protect the tooth surfaces You have to also protect the underlying connective tissue. Okay, just the selective interchange with the oral epithelium means there is a selective or semi permeability in the epithelium. Okay, now they have said this is achieved by proliferation and differentiation of the keratinocytes. This proliferation and differentiation of keratinocytes now has been divided. So now coming towards the proliferation. Proliferation means the division of keratinocytes will take place by mitosis, which occurs in your basal layer. We all know. Then it has less frequently in the suprabasal because they are mature. Okay, and the small portions will be left in your basal layer. Okay, because large amount of cells will be migrated to the superficial layer or surface of the cell. Okay, differentiation, which is basically known as the process of keratinization. Basically it consists of physiochemical and morphological events, okay, that occurs during when the cell migrates from the basal layer to the surface layer. The main changes is your flattening of the cell. As you all know, as you all know, you can see here the diagram. This is the tall columnar cells. Then you have little bit cuboidal. Then it becomes flat. While moving or differentiation, it will move into the flat cells. Okay, you should know the structure of the epithelium. Okay, you should know the structure of the epithelium. It's very important. Okay. Okay, so you should also know that in which cell the proliferation occur. It is basically the basal layer. The differentiation will occur when it moves, when the cells migrate from the basal layer to the surface layer. And what changes are basically morphological. Number one, it is basically the number one, we have said that it is the flattening of cell, but also with the increasing of prevalence of tone of filaments. Next, we have hyaline granules. In these granules, there will be keratin. The keratin will be when it moves to the superficial layer, it will convert it into a mature form, and then it is also disappearance of the nucleus. Then further, after the complete process, you have two type of nucleus. One is orthokeratinize, the other is para. After complete keratinization process, it will lead to a production of the orthokeratinize layer. Always remember, in ortho there is no nucleus, in para there is nucleus. How can you remember this? P means present. So you can say that in the parakeratinized the nucleus is actually present. Okay. So the orthokeratinize is a superficial horny layer, which is basically similar to the skin layer. Okay. Okay, but has no nuclei, as I have said, in the stratum cornea, and it has a well defined stratum granulosum. Okay, and only the some areas of outer gingival epithelium it is ortho. The rest is covered by a parakeratinized or non-keratinized epithelium, which consider an intermediate stage, and this stage will progress to maturity when there is any pathological or physiological condition. Okay, in parakeratinized there will be pyknotic nucleus. You should remember also keratohyaline granules that are dispersed rather than it will be staying or giving rise to the granulosum layer. Okay, in non-keratinized, always remember there is a cytokeratins, which is the major component in all epithelium. The major component in all epithelium is cytokeratins. Okay, it has neither the granulosum layer or strata coronium coronium strata, whereas superficial cells have viable nuclei. Okay, it's very important that you should remember. This is the four layers of the keratinized epithelium. You have stratum basilia, spinosum, granulosum, and corneum. In your non-keratinized layer, there is no stratum granulosum layer. Okay, it's very important to remember there is no stratum granulosum layer you have. Okay, basically, yes. Okay, Moving towards what I have said to you, that we will also study about the non-keratinocytes layer. Number one is melanocytes. Melanocytes, as you know, that these are the dendritic cells, and the dendritic cells which is present in your either basal layer or spinosum layer of the gingival epithelium. They synthesize the melanium in the organelles, which is known as pre-melanosomes or melanosomes. It's very important. You should remember the cell, the location, and what its function. Langerhans cells are basically among keratinocytes at all. It is basically in suprabasal layer, means it is present in the basal layer but in their supra level. Okay. It is a mononuclear phagocyte system, matlab it is a reticular endothelial system. Okay, you have it is a modified monocyte that is derived from bone marrow. Basically they are present in your bone marrow, and these are the cell signaling. How? Because it is antigen presenting cells. Yes, they are considered a macrophages with the possible antigenic properties. They have antigenic properties. They present the antigen to the lymphocytes. Okay, they contain barbic granules, and they have also adenosines. They have ATP activity. Okay, they are found in the oral epithelium, okay, of the normal gingiva, and small amount in the they are in the oral epithelium, also circular epithelium, and they are probably absent in junction epithelium of the normal gingiva. Always remember, Langerhans cells are absent in the junction epithelium. Okay, Marker cells is basically present in your deep layers of epithelium. They are nerve endings, so you can say touch receptors. Okay. Okay, touch receptors. Okay. Okay, one thing, they are basically connected by desmosomes. Yes, as you have studied in the previous table, there are some cell to cell connections, hemodesmosomes or desmosomes, etc. Okay, and there are tactile receptors. You know that basically marker cells, all nociceptors, you know, you have studied in your first second year basically. Okay, the epithelium is connected to epithelium is connected or joined by underlying connective tissue of basal lamina. Basal lamina then have lucida and densa. Always remember, then the densa will be towards connective tissue. How you will remember? Then densa is towards the connective tissue. Dense, malab connective tissue, it looks like a dense, that it has a lot of things if you consider it. Dense and lucida is towards the epithelium side. And basically, hemodesmosomes of basal epithelial cells is like towards lucida, which is composed. The lucida consists of the glycoprotein laminin. Okay, and the densa, which is basically a collagen 4, and basically a collagen is also present in your connective tissue. Basal lamina, which is clearly distinguished at the ultrasound level, it is connected. Basically it has condensation of the underlying connective tissue fibers, like collagen 4 is present in here. Okay, the connective tissue fibers by the anchoring fibers. Okay, like this. Anchoring fibers have been measured, like they have, it is not important. Okay, coming towards it is most important. The complex basal lamina fibrous is the periodic acid Schoffen positive and uh, Philip line observed in the optical lever. You should remember the basal lamina is permeable. Yes, it is permeable to fluid, but it acts as a barrier to particular matter. We have studied that the oral epithelium is basically selectively permeability. It comes like the basal lamina is permeable to fluids, but it acts as a barrier to a lot of things. Okay, so now we have discussed the microscopic features, and we have discussed the cells. The major cells were keratinocytes, the others non keratinocytes were Langerhans cells, marker cells, and also we have melanocytes. Okay, if you have any query, please tell me in the comment section. Thank you.